Welcome. We are slowly approaching the end. We have finished discussing all the cells that you are going to find in a plant. We are now going to talk about the organs. There are two organs we're going to talk about, and that will be the xylem, and we're going to talk about the phloem. The xylem of the two, the xylem is the most complicated one. I didn't say it's difficult, I said it's complicated. And the phloem is much easier. Okay. Xylem, or the plant body, comes in two forms. The form that comes straight from the embryo and grows, and this is often the form that grows vertically. And after a while, plants like human beings stop growing vertically and they start growing horizontally, becoming bigger and bigger. That is called secondary growth. So we're going to recognize primary xylem and we're going to recognize secondary xylem. Luckily for us, once we've understood primary xylem, there's very little to add for secondary xylem. Okay. Now, xylem is the water conducting tissue of a plant. No other system conducts water except xylem. Therefore, it is only xylem that has got cell group number 1. Cell group number 1 is called tracheary elements. Then you are going to say, we never studied those cells. They are not cells. They are a group of cells grouped together because of the function they do. These cells are there only for conducting water. Tracheary elements refers to two cell types, the tracheids and the vessel members. The tracheids are those elongated cells which I said look like fibers, but these cells are imperforate. Imperforate in botany doesn't mean without holes. There are holes on the side, and those holes on the side of the cell are called pits. When we say a cell is perforated, it has special wide openings, normally at the terminal regions. So the tracheids are imperforate. They don't have these openings. The vessel members are cells. When you say members, you're talking about cells. The vessel members are cells that are perforate. That means they have holes at the top and at the bottom, big holes. They also have pits, but that doesn't make them perforate. It is the big holes that they have at the terminal ends that makes them perforate. Now, a vessel member is a single cell. When vessel members join end to end through their perforations, then we have a vessel. When it is one, it is a vessel member. When they're joined and form a pipe, then the pipe is called a vessel. So if you're supposed to talk about cells of the xylem, you talk about a vessel member, not vessels. So when you talk about tracheary elements, you're talking about the tracheids and the vessel members, both of which are non-living. They are dead. They don't have a nucleus. They have a big hole in the middle. So their main function is to conduct water. We have another group of cells which we call fibers. Fibers may be dead or they may retain protoplasts. Fibers are here to support the xylem. to make it stronger. They are also here to store food. Very often they will store food if we don't have enough parenchyma cells. Fibers are cells, and these fibers are of two types: fiber tracheids and libriform fibers. A fiber, we know, is a cell. A tracheid is a cell. But when we say it's a fiber tracheid, we are describing a fiber, not a tracheid. If we say it's a libriform fiber, we are describing a fiber. These two will vary by their lengths. These cells are needle-shaped cells. They are imperforate, but they do have pits. They are mainly for support. Parenchyma cells are living cells. They will store food. They will pass products from cell to cell. They will get rid of wasted, unwanted matter, which is gastric substances. And they help in overall movement of water from the xylem to the rest of the other cells. So there are three types of cells, and these are the three types. In primary xylem, I told you, we are talking about xylem that goes normally vertically, growing in height. Hence, primary xylem is said to have an axial system. Vertical growth, axial system. Secondary xylem, on the other hand, has two methods of growth: an axial system, which takes care of growth vertically, and a ray system, which takes care of growth horizontally. The ray system has parenchyma cells, and it may have tracheids in gymnosperms. Okay, let's look at xylem. We know its function. It is to conduct water. We have already classified it as the primary xylem that comes from the embryo and from the apical meristems, the shoot and root apical meristems growing vertically, and secondary xylem that comes from the vascular cambium. Okay. Primary xylem can be separated into two. This vertical growing xylem can be separated into two: protoxylem, first formed xylem, and metaxylem. This is just dividing them by when they came into being. But we will note that if we had done a practical and we had looked at xylem, protoxylem, normally the cells have a small lumen. That means they have a small opening. Whereas metaxylem, the cells have bigger openings. The reason why these have a smaller openings is because as metaxylem is produced, it pushes this one, and this one becomes squashed, and the lumen becomes smaller. Okay, now I told you tracheary elements are two things: tracheids and fibers. Let us look at these tracheary elements, how they develop. Okay. The secondary thickening of the wall of tracheary elements is not haphazard. It doesn't just happen any odd how. The way the secondary wall is laid down gives us a variety of patterns. Please go to your books and look at the diagram we have on page, oh, where is it now? On page 54. On page 54, at the top, you will have a number of diagrams, and these diagrams illustrate these patterns. If you look at the patterns, you will see that when it is an annular pattern, we only have the thickening in a ring, and in between we have a thin wall. When it is helical, the thickening is a little bit more because it is connecting two rings together and going on to the next. When it is scalariform, the rings are tighter, and when it is reticulate, it is so tight that we no longer can see the rings, but we just see a net. And when it is pitted, we find that the whole wall is solid, it is hard, except for the holes that remain on these walls. Now, when you have a tracheary element, you will know that you are looking at protoxylem by looking at, from the side, from a longitudinal section, you will see either annular or helical thickening. If it is metaxylem, you may see some helical thickening, but mainly it will be scalariform, reticulate, or pitted. However, if it is a transversal section, it is the only way you can separate protoxylem from metaxylem is from the size of the lumen. Small lumen, larger lumen. I told you that perforations are different from pits. Pits are normally on the side of the vessel member or the tracheary element, but perforations are terminal. This is a simple perforation. It is just one hole. This is a scalariform perforation. It is like ladders. There's material in between the holes. And then here we have reticulate perforation, where we can just see a net-like perforation. These are the perforation plates, and on the side we have pits, in botany not perforations. Then we are looking at fibers. We said fibers are for strengthening, they are for support. There are fiber tracheids, they are libriform fibers. Fiber tracheids, now you can't say this is a fiber tracheid or a libriform fiber. It is a relative term that, given a lot of tracheary elements, those that are shorter and thinner with bordered pits are fiber tracheids. Those that are thicker with slit-like pits are libriform fibers. Fibers, regular fibers, will be anything, will be any combination of these. So if you don't know if it is a fiber tracheid or a libriform fiber, just call it a fiber. That way you are fine. Now with fibers, we do have, in very rare cases, what we call gelatinous fibers. That means they look like fibers, but they are not hard. These are found in what we call reaction wood, and this is the wood that will be in the pulvinus, or where the branch has to be able to bend. Okay. Fibers, when they do retain protoplasts, like I said, sometimes they will be for storage. When they do retain protoplasts and they're going to store, we will find that these fibers are often septate. That means they have divisions within themselves in the axial mode. Okay. Right. When we talk of the vascular system, we are talking about xylem and phloem. I've presented cells of the xylem. You will find that in most cases when you look at xylem and phloem, there are certain things that are common. Parenchyma cells would be common. And for support, whereas in the xylem we have fibers only, these two types, in phloem we have fibers and sclerids. In short, we have sclerenchyma. We don't have sclerenchyma in the xylem, only the fiber component of the sclerenchyma. Now, I said the tracheary elements are unique to xylem, just as the sieve elements are unique to phloem. When we talk of sieve elements, we are referring to, we can say 2 cells, but actually it is 3. Sieve cells, sieve tube members, and companion cells. However, sieve tube members always go with companion cells. Companion cells are not the same as sieve tube members, but they always exist with sieve tube members. Sieve cells are something else. Okay. I've already told you that primary xylem is the xylem from the embryo, so is primary phloem. Protoxylem is the first formed one, metaxylem is later formed one. Please note that in phloem, you seldom have protophlloem and metaphlloem existing together, because the cells of phloem are soft cells. When metaphlloem comes out, it pushes protophlloem, and because protophlloem is soft, it is destroyed. So in secondary phloem, we only talk of metaphloem. They seldom exist together. Secondary xylem, I've also already told you, comes from the secondary meristem, which is the cambium. The secondary xylem comes from the cambium. This is a section of the root to show us the location. I told you that if it is protoxylem, the lumen is small. If it is metaxylem, the lumen is big. The lumen is just the holes that you see here. So this is later formed, this is first formed. But when it comes to phloem, we don't bother about first formed or later formed. We just normally just talk about secondary phloem, primary phloem. We don't separate it. Now, let me mention this area here, which is the area with pericycle. This is the cortex. Then we have the endodermis here. Then we have one layer of parenchyma cells, which we call the pericycle. The pericycle is important in the root because this is the source or the origin of secondary roots. Secondary roots, we normally see them out here. They don't start from the epidermis. They start from the pericycle and grow out. Hence we say they have an endogenous origin. Okay, thank you.